And Gary, if you want to go ahead and share your screen.
Will do.
We'll get started.
There it goes. You ready for me to go ahead and jump into it then?
Yeah, let's go.
All right. Well, hello everyone. Thanks for coming to hear the talks. I'm Gary ZeRuth. I'm a professor in biological sciences at Murray State. And today I want to talk to you about one of the projects that I've been working on in my lab. And this project that we are working on kind of centrally revolves around a particular transcription factor that we identified in my postdoc lab. So when I was a postdoc at the NIH, we identified through a yeast-2 hybrid analysis these transcription factors that we named Glis-similar because they share a lot of homology with the downstream effectors of sonic hedgehog signaling, the Glis-family of proteins. And these transcription factors are kind of broadly expressed throughout multiple systems in the animals. So we decided to investigate what their function was. And we found through making knockout mice that loss of the transcription factor Glis3 led to a multi-system phenotype that included polycystic kidney disease, neonatal diabetes, hypothyroidism, and a whole host of other things. Since that time, there's been upwards of 20 genome-wide association studies now that have implicated Glis3 mutations as a risk locus for the development of type 2 diabetes. So given the prevalence of diabetes, we in my lab have been really focusing on the role of Glis3 in the development of the pancreas, as well as how dysfunction in Glis3 can lead to diabetes. I don't think I really need to go over the data. Everyone's aware that diabetes incidences have been increasing pretty steadily over time, not just in the United States, but also globally. In the United States, we're now looking at the most recent CDC data, has about 29.4 million Americans with diabetes, or just under 10% of the population. So diabetes is a disease that affects the ability to properly regulate blood glucose. So at the heart of diabetes lies the pancreas, which is the organ responsible for regulating blood glucose. The pancreas is made up of three major types of tissue. You have the acinar cells or the exocrine cells, and their job is to produce digestive enzymes, particularly amylase and trypsin and chymotrypsin to digest fats and carbohydrates in the diet. And then you have your ductal cells of the pancreas, whose job really is to transport those digestive enzymes from the exocrine portion into the duodenum of the stomach. About 1 to 5% of the pancreas by mass, depending on the species you're looking at, is made up of these endocrine cells, and their job is to produce the hormones that help regulate blood glucose. And these endocrine cells are organized into islets. This is a zebrafish pancreas we're looking at here. You can see the vast majority of the islet is comprised of these insulin-producing beta cells. And then you have a population of these glucagon-secreting alpha cells that kind of flank the periphery. And then your somatostatin-producing delta cells and pancreatic polypeptide makes up a small minority of the islet. So diabetes occurs when the system does not work properly. So under normal conditions, when you have elevated blood glucose because you just had lunch or whatever, then the elevated blood glucose is going to trigger the endocrine portion of the pancreas to secrete insulin into the bloodstream. Insulin then binds to the peripheral tissue after circulating through the bloodstream and binds to the insulin receptors, which then leads to an opening up of the glucose transporters, allowing your peripheral tissues to take the glucose out of the bloodstream and use it to make energy. So we use the zebrafish as our model for tracking pancreas development and function. And they're a great model for this because their pancreas develops largely in the same way that you would see in humans, but much more rapidly. And the embryos are transparent during development, which allows us to visualize these processes under a microscope. And fortunately, the zebrafish is also really amenable to genetic techniques, which allows us to do overexpression and knockout models with relative ease. So the development of the pancreas within the zebrafish kind of starts around 24 hours post-fertilization, where you'll see a dorsal bud emerge off of the gut tube right below the stomach. And then by about 32 hours post-fertilization, you get this ventral bud emerge. Now, the dorsal bud in zebrafish is made up entirely of endocrine progenitor cells that'll eventually turn into the endocrine portion of the pancreas. The ventral bud is a mixture of all three lineages, so you'll have progenitor cells for the exocrine, ductal, and endocrine cells in there. By about 2 days after fertilization, the ventral bud is going to rotate around and fuse with the dorsal bud, engulfing it, and then undergo a rapid period of maturation and expansion, giving rise to the pancreatic body and the intrapancreatic duct. And then by about 5 days after fertilization, you'll see secondary islets start emerging along the pancreatic duct. And these are the islets that are primarily responsible for making the insulin to regulate the blood glucose in the adult organism. So we have been tracking in my lab the spatiotemporal expression of the Glis-3 transcription factor during early development in the zebrafish. And we find for the first 2 days of development, we see Glis-3 expression seems to be limited to the brain and the kidney regions. And we don't really see Glis-3 expressed in the endoderm until about 72 hours post-fertilization. And by this point, we see that Glis-3 is pretty broadly expressed through the endoderm, including distinct expression in the pancreas. So we wanted to see what effect knocking down Glis-3 would have on pancreatic development in the zebrafish. So we employed a splice blocking morpholino that binds within the Glis-3 locus right in between exon 3 and exon 4. And this should prevent proper splicing to cause the entire deletion of exon 4, which is made up of 589 base pairs. This is going to result in a frame shift mutation that should knock out Glis-3. So when we do this, we get about a 40% reduction in Glis-3. This is primers that are within the Glis-3 exon 2, and you see there's no major difference. But when we use primers specific to exon 4, we see about a 40% reduction. And we also quantified that by RT-PCR and showed again about a 40% reduction in Glis-3 expression. And this resulted in about a 25% reduction in all three of the major hormone producing cells. So insulin, glucagon, and somatostatin expression was down by about 25%. And we get a 50% reduction in some of the major transcription factors involved in specification of the pancreas. This is pancreatic transcription factor 1A and PDX1 were dramatically reduced. We have fishes that report insulin. They have M-cherry downstream of the insulin reporters. We can actually visualize insulin expression in the fish. And we see that the morpholino against Glis-3 doesn't really result in apancreagenesis, but the expression of the insulin signal is much more restricted to a smaller area, suggesting to us that Glis-3 either has a role here in the specification and maintenance of the progenitor cells in the ventral bud, or in the expansion and maturation of the cells after they fuse. But since morpholinos typically are only effective for the first 3 to 5 days after injection, those later time points in development become difficult to study with Glis-3 knockdown. So we use CRISPR-Cas9 technology to try and make a knockout line where the fish permanently do not express Glis-3. So we designed 2 guide RNAs that both will induce a double-stranded break within exon 4 of the Glis-3 locus, and if both simultaneously recruit Cas9 to cut the DNA, we should be able to excise 179 base pair fragment from exon 4, resulting in a frame shift mutation. So we've injected the fish with this and resulted in about a 66% cleavage efficiency. So we're now growing these fish up to analyze to get founders so we can get a stable Glis-3 knockout line. So aim 1 of the grant that we're currently working on then is to evaluate these knockout fish that lack Glis-3 expression, so we can kind of determine where Glis-3 might be acting in the pancreatic developmental paradigm and see whether it's involved in the specification and maintenance of progenitor cells, in the expansion and maturation of the ventral bud, or in the formation of secondary islets that emerges later. Aim 2 of our grant is looking at susceptibility of the fish to type 2 diabetes that lack Glis-3. And type 2 diabetes doesn't occur initially due to a failure to produce insulin, but in insulin resistance at the peripheral tissues. So the current model for type 2 diabetes is peripheral tissues get insulin resistance that makes them less responsive to the insulin molecules. And early in the disease, the organism compensates for this simply by increasing insulin expression to make up for that. But for reasons we don't completely understand, they then enter a period of dysfunction where insulin levels rapidly plummet, causing blood sugar to rise and resulting in diabetes. So we tried to make a type 2 diabetes model in the zebrafish by feeding them high-fat, high-glucose diet. And after about 30 days, we have found that they have extremely high blood sugar. Their fasting blood sugar is elevated relative to the control fishes. And that is coincident with a severely increased amount of insulin expression. So we think these fish are somewhere here in the early states of dysfunction where they have elevated insulin and elevated blood sugar. So we want to examine the Glis-3 knockout fish under this high-fat diet model to see if it makes the fish more susceptible to type 2 diabetes. And finally, our third aim is looking at zebrafish have a relatively unique ability amongst animals to regenerate beta cells after ablation. So you can chemically ablate all of the beta cells in the zebrafish, and they will regenerate them in about 2 weeks without any need for insulin intervention in between. So here you can see our model. We inject streptozotocin into a zebrafish. This is the pancreas that we're observing here with the red signal. And by 2 days post-injection, all of the beta cells have been completely ablated. And then by 17 days post-injection, we see that signal starting to return here while the beta cells regenerate. So what we would like to do is use our Glis-3 knockout model to chemically ablate the beta cells with streptozotocin and then analyze the different steps of beta cell regeneration to see whether Glis-3 is required for the beta cell pool to regenerate, or whether the early embryonic development of beta cells and regenerative pathways work under different mechanisms. So that is all I've got time for today. But I would like to give some acknowledgement to my lab, particularly my graduate student, Tasha Sweeney, who's been instrumental in doing all this and helped me collect a lot of this data. I also have a talented group of undergrads who help with this work. And I'd like to acknowledge my funding. I have an NIH R15 grant that helps pay for all this. And I also like to thank the Kentucky INBRE for all of their support. And I can entertain any questions if you have them.
Thanks, that was great. Any questions for Gary? Another. Nothing's coming to mind right now because it was all very clear. Any other zebrafish people?
Actually, can I ask a question?
Sure.
So Gary is completely out of my bailiwick, but I'll try to come close here. What happens if you overexpress Glis, or people looked at that? How does that affect all these different parameters you just talked about?
No one's looked at it to my knowledge. We have been desperately trying for months now to overexpress a green fluorescent linked mRNA to see what happens. And what we expect would happen, or at least we were hopeful, that overexpression of Glis-3 would cause expansion of the pancreas into regions it doesn't belong, like into the stomach and into the intestine. Unfortunately, we can't get it to work. So I have a student working on it this semester. Hopefully we'll know soon, but to my knowledge, no one's ever tried overexpression.
And actually, I have one more question. I remember I had another one. What's the viability of these fish like? Are there any, is there a phenotype? Do they live just as long as the other fish, or with the Glis knockouts?
No, that is a fantastic question. So when we knocked this out in mice, they died at PND5, most likely because the mice had no beta cells at all. They were completely wiped out. The fishes we thought would be a much more viable model because they can survive without insulin or with reduced amounts of insulin, unlike the mice. And it doesn't seem to be as involved in the specification of the beta cells. So when we initially made our group of founder fish for the CRISPR, they all looked relatively healthy and happy for the first 14 days, and then we had a massive die-off. So we think it may be lethal at that point. So we might have to generate heterozygous lines and just breed them as we go. So we're not sure. We're repeating it again to see if we get the same type of die-off. We know Glis-3 is expressed in the sperm, and it looks like it has an effect on the Sertoli cells. So there might have a reproductive defect as well when we knock it out in a homozygous way.
Thanks. That was a great talk.
Yeah, thanks, Gary. I guess we should move on to Dena. You want to share your screen? Yeah, sorry, you're muted, Dena.
Sorry, weird things happened. Wasn't supposed to do that. We'll just try it again.
Yeah, it was shared nicely a minute ago.
Yeah, and then craziness happened. So
Yeah.
we gotta get it back. We'll get it back. Okay. Now we're good. All right. Hello, I'm Dena Weinberger. I'm an assistant professor also at Murray State, and
I don't see your screen, do you?
Oh, man, I'm in trouble with this. We're just going to end the show and try again. Okay.
Guess we can't have slides and volume.
Can you see
There
it?
we go. Yes.
Yay.
Yes.
Okay.
And I can hear you.
We can hear me. We can see me. Everything's good. All right. So I also work in Zebrafish. My title, a dual regulator of cerebellar development and function with a question mark. The goal of this is to remove the question mark. This is based off work I started as a grad student. And so these are some old pictures where you're looking at the gene expression in little baby fish. And we're focusing on this little region right here, which is the cerebellum. And so I analyzed other things with my graduate work and left that and decided to come back to it. So just to give you an overview of what my lab is focusing on, my research goals are to study mechanisms of development and more specifically to use that type of mechanism as a basis for someone else to look at foundations for early interventions. So since I'm focused on the nervous system, what are things that we can do when we know there's a problem likely because of, a genetic defect or whatever, to minimize that risk or to devise therapies when early mitigation isn't a possibility. So that's the framework that I work through. And I focus on the development and function of the motor system. And today I'm going to be focusing on the cerebellum, which is this region in the zebrafish. There's a really cool foliated structure in us and rodents that has the most famous roles of coordinating motor activity and controlling motor learning. It has this very stereotypical anatomical structure where we have layers of excitatory cells and layers of inhibitory cells, and then a layer where they're all making their connections. And the zebrafish has obviously a much more simplified version of this mouse one right here, but it follows the same patterns. And also what's cool about these populations of cells, aside from them being the most abundant neurons in your brain, these little excitatory cells, these are normally overproduced, way overproduced, and then they're pruned to make sure that all of the appropriate connections are made. It's also associated with a certain type of pediatric tumor called medulloblastoma, where there is a failure in either the overproduction stopping or a failure to prune away these cells, and you end up with a cerebellar tumor. My interest is on this protein called cabin-1, which is known to be a multifunctional repressor protein that is primarily known for regulating phosphatases, but also transcription factors. And there's this transcription factor that is known to repress called MEF2 that is involved in the generation of these granule cells, that excitatory population that's overproduced. MEF2 produces or stimulates the production of pro-survival factors. If you don't have it, then these cells do not survive very well. Cabin also is known to regulate another transcription factor, P53, the tumor suppressor, which is more famous for promoting death. So that when P53 is active, we get the production of pro-death factors, which then would be presumably responsible for removing this population of cells. So already you can see that there's a possible conundrum because cabin represses the one that makes cells survive and it represses the one that kills cells. So there's kind of these different options, and either is one possibility happening in a time-dependent manner? Is it repressing both of them in different cells? Is it not involved at all? These are all possibilities that ultimately cabin could be regulating the population of granule cells by altering the survival of populations of cells or altering this, the killing off of those cells. One of the advantages of working in the zebrafish for this specific question is that the null mutant of mice is embryonic lethal. So there's not a whole lot known about what cabin does in the mouse, and certainly in a developmental context because of this. My hypothesis for this section is that cabin is playing a role in the development of the cerebellum granule cell population through some sort of interaction with MEF2 and or P53. And there's going to be some sorting to see which one is involved in the future, or both. So what I'm looking at is to see if I knock down cabin using morpholinos, so that I got a total reduction, but not a 100% elimination. What's the effect on proliferation and cell death, which I'm looking at using phosphohistone 3 staining to mark proliferating cells, and then tunnel assay to label apoptotic cells. So these are going to be labeled with different colors. I'm just showing you the green ones today. That would be our proliferating cells. So we're labeling for phosphohistone 3, which is indicative of dividing cells. These are sections through the zebrafish brain at 4 days post-fertilization when they were injected as eggs. And we're tracing boundaries of cerebellar tissue. And then we're analyzing, instead of counting cells, because this is such a dense cell population, they're tiny and they're very densely packed here, we're using instead intensity labels to look at the area of the tissue that's labeled. And this is still ongoing, so I don't have final numbers for this yet, but it looks like in general with cabin 1 morpholino, this is a translation blocking morpholino, we get a reduction in the number of proliferating cells. I've also got a little bit of data with a different morpholino that's a splice blocker where we see a similar trend. And I'm in the process of acquiring the CRISPR-Cas9 mutants that were developed by my previous lab in conjunction with another one. And literally, I'm just waiting on the legal issues right now of transferring them. So soon, and then I can look at these as well. But what it's looking like from these preliminary experiments with the morpholinos is that possibly what's happening is that when we lose cabin 1, we get not as many proliferating cells. And so maybe there's more of them that are dying instead. So I don't know the answer for sure on that yet, but it's at least interesting to think about. And we're working on kind of the next steps in refining this. And also, we don't know yet what's happening with these cells that are dying, that with the tunnel stain. But there might be some clues about which population is being affected by looking at which cells are actually expressing cabin 1 rather than the whole cerebellum itself. So I'm also looking at which of these major cell groups is expressing cabin 1 using double in situ hybridization, where we stain subsequently with different colors. I'm using one color for cabin 1 and one color for a marker. In this case, it's ATA1B, which labels the granule cell precursors. There's also a lovely transgenic line, so you can see that it makes these cool populations that migrate and radiate at 1 day old. And with our purple here labeling ATO1B, cabin in red, or in this place, cabin in just purple, since that stain is a little more clear, we don't see that expression. So it looks like cabin is not involved in the cerebellar granule cell precursors, although we do still get that expression in these regions of the cerebellum. It just doesn't seem to be in that specific population. We're in the process of looking at another marker that has been riddled with complications, which incidentally is also one that Gary mentioned. PTF1 alpha is a pancreas transcription factor that's also involved in the specification of the inhibitory cells of the cerebellum. Another question, though, that I wanted to get at was, I knew that cabin was expressed in the developing cerebellum, but the cerebellum is actually one of the last parts of the brain to finish developing. It's pretty slow. It's really complicated. So I followed that up through looking at just in situ hybridization with single and doubles in the juvenile stage and in adults to see what the late maturation stages of the cerebellum, which would be approximately 1 month of age. And then in the adult, where we're only looking at the limited neurogenesis and the plasticity of those cells. Otherwise, that's where we know that MEF2 and P53 might be playing roles in those cell populations. So this was done on section tissue because their brains are too thick otherwise. So just to orient you, here's anterior, posterior, dorsal, ventral. What's important here is this region that's zoomed in. This is the cerebellum of the zebrafish, where we have the granule cell layer, the excitatory cells, the Purkinje cell layer, which is the inhibitory cells, and then this molecular layer is where all of their axons and dendrites are. And cabin is in brown. This is using a different marker that's known to label the Purkinje cells in purple, and we see basically no overlap. However, since there's that really discrete anatomical marker, we can also be assured that not only is it in the granule cell layer, which also extends out here and a little bit in front, it's absent from the Purkinje cell layer. And this is in accord with what was seen in the rat several years ago, where cabin is known to be expressed in specifically in the granule cell layers of the rat cerebellum. And then we also saw some interesting similar expression patterns throughout the whole brain, including in the olfactory bulb, the hypothalamus, and then regions that are known to be part of the memory type areas analogous to the hippocampus. So this is exciting because now we know what we're doing in the zebrafish matches what we know about cabin 1 in the rodent as well. So that's always a good thing to have in store, since ultimately the goal is to have things make sense for what we would do in humans. We also learned that in situ protocols vary a lot, and what works for one gene doesn't necessarily work for all the others. And even though I had lots of in situ experience as a grad student, we've run into so many problems. So this became a project for an undergrad who was working on this and running into all sorts of difficulties. We've tried other people's protocols, and everyone's got their tips and secrets and tricks and magic that they wave their hands around and say, so we tested them. We looked at different additives that are supposed to make clearer signal, which we would absolutely agree that dextrin sulfate is the winner here. We now use it all the time. There's also a lot of variation and difficulty in the double in situs, which are very difficult. Each of these is showing a different stain pairing of the exact same things. So in purple here, or in purple here, or in this darker blue here, that's all ATO1B, that's the granule cell precursors. In brown or in red or in light blue is cabin 1 expression. We can see the sense probes on the bottom, just so you have a rough idea of what it looks like and what the background looks like. So we are using these trials and errors into an undergrad publication. So comparing these different protocols, putting together everyone's magical stories and getting something useful out of it. So we are in the process of that. We're aiming for early submission next year. I wanted to give an update on the grant status. So the first time I submitted this in 2017 wasn't scored. Big thing was more data. So I got more data. The next year it was scored. One of the things they wanted was more publications. So I got more publications. And then I resubmitted and it wasn't scored. So I'm still working on this and I will resubmit it. But I think part of it is, and you know, from filtering all of the comments, they want to see mechanism and they want to see evidence that what I'm proposing is actually going to work. And so I think once I get some of this first stuff published, then I'm going to see a little bit more positive reaction. And I also think that when I can get some preliminary data with the CRISPR fish, that will cement my story a little bit more. So it's happening. We're going to make progress. But I also wanted to highlight that because this has been very difficult and very slow going and not successful on the grant front, I have other projects that have been way more successful. So this is just going to be super quick for all of them. So I can say, yay, look, wonderful things are happening. My postdoc work got published in 2020 in PNAS, which was looking at, again, motor system and development of the neuromuscular junction. That has led to a project that is ongoing in my lab that's on hold right now because of COVID stuff, but we'll get back to it. But the early parts of that extension led to a collaborative paper with Gary ZeRuth, looking at methods paper in zebrafish for immunohistochemistry. The novel piece of information there was that the zebrafish muscle also expresses NMDA receptors, which are glutamate receptors and not normally found at neuromuscular junctions. That also came out in 2020. And another collaboration with another Cabrin-supported person in chemistry with Dr. Subedi. We had 2 papers come out looking at the effects of drugs that are found in wastewater on gene expression and looking at how that affects zebrafish development. And this is leading to my next R15 proposal that I'm hoping will be a little bit more strongly supported by the NINPS. I've had a lot of undergrads working on this project. I've been funded by, Cabrin, now INBRI, and always thank you to the zebrafish. And do you have any questions for me?
It's great. I was just curious, are there fluorescent labels for the in situ that you could use for your double labeling, or is that
So
possible?
some of them are fluorescent and not fluorescent. And so meaning like they've got the chromogenic substrate and then they also have a fluorescent tag in them. I have one of them. I haven't looked at the fluorescent signal from it yet. The main problem with those is that they tend to work really well in tissue sections and not as well in the whole mount.
Oh, yeah.
So that seems to be kind of the general case overall, is that the double in situ is a lot easier in the tissue section where everything's really thin, because there's so many steps where we're washing and we're using all these harsh chemicals that ultimately it ends up kind of stripping off the first one a little bit. So that's part of the refinement, but that is an option. And I have a red one that I can check out. And there is also a blue one, as long as we have the right filter to see it.
Well, the other comment I might make is there's a new mechanism called the R16. I don't know if you had heard about that. It's just something to potentially explore, because it seems like it's even more money and it's really tailored to student involvement. So something to keep in mind.
I'm hoping so. Thank you.
Any other questions before we move on to Emily? Thanks, that was great. Okay, Emily, you want to try and share your screen?
Okay, can you see that? Okay. Hi, everyone. My name's Emily Shifley, and I'm at Northern Kentucky University, and I'm going to talk about my research on the role of FGF signaling during Xenopus Pharyngeal Development. So I'm a Xenopus lab. I love frogs. They're nice because they lay so many synchronously developing eggs for us to do cool experiments with. So we do a lot of similar stuff as zebrafish. We inject morpholinos to knock genes down, and we can inject RNAs as well. And then over here, I have some cool, you can do cool transplants of tissues with Xenopus embryos, and they just kind of keep developing. So I can inject a fluorescent lineage tracer and then dissect out a population of cells. So in this case, it's some neural crest cells, and then let those embryos continue to develop and see those neural crest cells have migrated into the pharynx in that neck region of the embryo. So before I talk about the research in my lab, I also just wanted to mention that if anyone ever has an experiment that they, need to do a transplant on or something like that, and are interested in trying it in the frogs, I'd be, open to collaborations. Sometimes it's more powerful if you can show that a gene's important in your model organism as well as another model organism. So if anyone's ever curious about Xenopus, then just feel free to contact me. Okay, so I'm studying the pharynx, which is an important structure in vertebrate embryos. This is just a little cartoon of it. So later on in development, the pharynx forms or contributes to a lot of different things. It helps form part of the craniofacial skeleton, some parts of the inner ear, some muscles and arteries of the face and neck, and then also the thymus and the parathyroid glands. Those are all derivatives of this pharynx. And if the development doesn't proceed normally, that can cause different kinds of birth defects. So these are just a couple birth defects that are known to be caused by improper development of the pharynx. So we'd like to know how the pharynx normally develops, and that can help inform how to prevent these kinds of birth defects. Another cool thing about the pharynx, so if we section through the embryo, this is a cartoon of it as well, is that you have multiple germ layers interacting. So the ectoderm is here in blue, the endoderm is in red, and then the mesoderm is in between them. So you have all three germ layers, and you have those neural crest cells that migrate into the pharynx. So a lot of different cell types congregate in this area to form all those different derivatives. So of course, all those different cells have to communicate with each other, and they use a lot of different genetic signaling pathways. And I've been focused on the FGF signaling pathway, which is known to be important for the development of the pharynx. So this is my little cartoon of FGF signaling. And of course, one effect of FGFs binding to the receptors is that you can have all different kinds of responses in the cell, but one would be gene expression. And then these are just cartoons of different stages of Xenopus embryos as they develop, and some of these different events in the pharynx. So at first, it has to actually form that architecture of the pouches and the arches from the endoderm and the mesoderm. And then the neural crest cells have to migrate in. And then there's a period where it's patterned and differentiated before the actual organ primordia bud out or, get formed. So that's at those later stages. So our first experiment, we just wanted to see, is FGF signaling important in Xenopus like it is in other species? for the development of the pharynx. So we cultured Xenopus embryos at different developmental time periods with a small molecule FGF inhibitor. And here's just a Western blot kind of making sure that our inhibitor worked. And then we looked at these embryos with in situ hybridization for a couple different genes that are known to be expressed in the pharynx. So this is just the head of a Xenopus embryo. And you can see a lot of these genes aren't only expressed in the pharynx, they're also in the brain and other areas. But these little stripes in the head region, that's the gene expression that we're interested in, because that's the pharynx of these embryos. And we saw some reductions in RALDH2, which is in the mesoderm, SOX9, and PAX1 is in the endodermal compartment of the pharynx. We saw reductions in PAX1, and TWIST is a neural crest marker. So we tried to look at a couple different markers and sort of quantify these reductions. And it seems like FGF signaling is actually needed in all the different time periods that we chose, but we got more severe defects between stages 23 and 27. And then we let some of the tadpoles grow to a later stage when they've formed their craniofacial cartilage. And so these are 3 different cartilages in the head that are formed by some of those neural crest cells that migrate into the pharynx and then help form these structures. And actually all the FGF inhibitions showed defects in this craniofacial cartilage, sometimes, kind of subtle here, or pretty severe, really small heads or completely missing some of the cartilage. So that was nice just to verify that FGF signaling is important for pharyngeal development. But to kind of add new information, we wanted to know what some of the targets of FGF signaling are. We know it's important for pharyngeal development, but there's not as much research on what genes it's turning on. So I did this experiment a while ago when microarrays were a thing, but we compared the early pharyngeal region of FGF inhibited embryos with control and got a list of downregulated genes, 2 of which are these Iroquois genes, so Iroquois 1 and 3. And so Iroquois genes are a group of transcription factors that were originally identified in Drosophila. In mammals, there's three genes in a cluster, so there's actually 6 Iroquois genes. 1, 2, and 4 are on one chromosome, and 3, 5, and 6 are on another chromosome. And they're known to play really important roles in lots of different areas of development: kidney development, brain patterning, limb buds, even heart development. But no one's really looked a lot at them and what they do in the developing pharynx. And there is one sort of rare recessive syndrome in humans that has a mutation in Iroquois 5. And they found using Xenopus that neural crest migration into the pharynx was disrupted. So my lab's been interested in trying to look at all the Iroquois genes, not just 5, and seeing what role they play in the developing pharynx. So we've been optimizing, knocking the different Iroquois genes down with morpholinos. So these are just control in situs, getting like a dose right, where they look like they're gastrulating okay at these early stages. So we're not just messing up the whole embryo. And then once we kind of got that dose right, we did more in situ hybridizations, looking at different pharyngeal genes. And some genes are unaffected. They seem pretty normal. Sprouty here is an FGF inhibitor, or snail is another neural crest marker gene. But CYP26A1, which we also saw being affected with the FGF inhibition, shows a lot of reductions with these Iroquois morpholinos. And then I submitted some of this as like preliminary data in a grant application, and the reviewers were like, I can't really see what's going on. So we've been moving to sectioning the embryos after doing the in situ, and it is much easier to see what's going on. because you can also see the arches and the pouches in the pharynx. So TBX1, we see maybe some expansions with Iroquois 3 and 5, and that's the gene that's mutated in DeGeorge syndrome. HOXA looked pretty normal here, but then when we sectioned CYP26A1, we could definitely see reductions in the gene expression, and also the morphology of the pharynx is a little bit messed up when we knock out multiple Iroquois genes. But another thing we noticed with a lot of these injected embryos is they have this kind of crooked appearance, which means maybe we are still messing up gastrulation a little bit. So I'm probably going to move to just injecting 1/2 of the embryo, and that might kind of help with that. So here's more in situ, is looking at some neural crest genes that look pretty normal when I just injected the morpholino on one side of the embryo. But with PAX1, which is marking the endodermal pouches of the pharynx, I could see reductions of the gene in embryos on both sides if I injected in both sides, and just the one side if I just injected it into one side. So I think, you know, sectioning the embryos and doing one-sided injections will kind of help our preliminary data look better and be more clear. And we haven't done a ton of this yet, but we also let some of the Iroquois knockdown embryos develop and looked at their craniofacial cartilage. And with some of the individual morpholinas, we definitely see like some slight defects. But when I knock out a whole cluster together, we saw more severe defects. So it's probably that they can play redundant roles. So if you just knock out one, the other one that's right next to it on the chromosome can compensate. And so I'll also continue to kind of knock out individual and combinations of the Iroquois genes. Okay, so what I've shown you is that my lab's looking at FGF signaling and kind of narrowed down a developmental time period when it's important for the development of the pharynx, and identified some different possible target genes of FGF signaling. And then we've been focusing on the Iroquois factors and trying to look at what they are doing during pharyngeal development. So we see some effects on gene expression and the morphology of the pharynx and with the craniofacial skeleton. And my future aims for the research are to continue to work on this project and address all the comments and suggestions from my last grant application. With the FGF experiment, you know, when we are inhibiting the small, you know, all the whole embryo, that's a pretty severe effect, but we have some different ideas for doing a more targeted knockdown or overexpression of FGF signaling with Xenopus to kind of see which germ layer it's important in. And then the Iroquois genes, another really important thing that the reviewers wanted to see was if I could rescue the effects of the morpholinos to know that it's specific to these genes. So I do have constructs for some of the Iroquois genes, mRNA constructs with a MIC tag. And so I've been able to express those, and it's just a matter of having the time to do the double injection of the morpholina with the RNA and hopefully rescue the gene expression in the craniofacial cartilage. Okay, so thank you to all the support I've had in the past from KY Embry and then all the different centers at NKU and all the undergrads in my lab. And I'd be happy to take any questions.
Thanks, that was great. We're a little bit behind, but are there some burning questions, or can we wait till the end?
Actually, can I ask one real quick one, Martha?
Yeah.
So Emily, is there a developmental time frame that CYP gene is expressed? Is it analogous to where you see the biggest effects?
Yeah. I mean, it's Yeah, it's expressed right at the right time for sure, but also earlier. Yeah.
Thanks.
Sure.
Okay, why don't we move on to Dae-Sung, and then, you know, there's time at the end. Hopefully you guys can all discuss.
All right. Am I on? Can you see me and can you hear me okay?
Yes, yes, you're not in presenter mode on your
All right.
slides.
Not presenter mode?
No, not yet.
All right. I am actually.
Oh, well,
There's something's wrong with my. All right. Swap
I mean-
presenter view like this? No? Is
there we
that it?
go. Good. Yep,
Okay.
perfect.
All right. Well, my screen it's still flipped, but it's still okay, I believe. All right. All right. All right. I'm Dae-Sung Hwangbo. I'm an assistant professor in the Department of Biology at U of L. Little bit of myself and my lab, because I'm only been here 2 years. I almost never met any of you pretty much. So my lab was established in August 2019, a little bit more than 2 years. Now I have 2 PhD students and 1 master student, but all of them are less than 1 year, very fresh. And I have 1 master non-thesis students just graduated in May. And I have 5 undergraduate students who are active, doing something with me. And I have a similar number, or kind of on and off, and different stages, like dormant stages, sometimes comes, sometimes on. My research named is Genetic Mechanisms of Dietary Restriction by the Circadian Clock in Drosophila, was funded by KY, IMBRI, IDEA. And I'm doing the 2nd year of this grant. I submitted R15 and got the score back. It's not really miserable, but I have to work on this to resubmit. I'm planning to resubmit in February of next year. I have a little bit discussion at the end of this presentation. So my research topics and approach, I use Drosophila, the fruit fly here. I just saw some of you use this, and many of you use, I believe, vertebrate, and my organism is invertebrate, very different. I used classical genetics and molecular genetics, and my topics, the first thing is aging. So if I have to choose a single word for my research, that is aging, but I'm trying to connect aging and circadian rhythms and sleep and feeding behavior all together. So for example, how aging process impacts on circadian rhythms and sleep, or vice versa. During aging process, what happens to your feeding behavior, or does feeding behavior change your rhythms and sleep? All are connected, and we're trying to dissect the mechanisms of interconnection. And in the future, we're trying to expand our research to Drosophila models of human disease, especially some neurodegenerative disease, including Alzheimer's disease. And one practical reason is actually for money. I mean, we are all in one team, I believe. So let me try to be honest. So one of the reasons is to bring more money, if possible. And that being said, I'm really interested in any type of collaborations with Caribbean researchers for publishable and fundable research. By the way, you can stop me anytime during my presentation if you have any questions, because my presentation will be just 10 minutes, I believe. So one topic of aging, my primary topic, is called diet restriction. This is very simple concept, very ancient concept. So basically, if you cut down your calorie intake or food intake, whatever you say, you're better off. Your lifespan extended, you're healthier. Everybody knows, but the molecular mechanisms are still up in the air. So for example, this is one of the old study in mice. Green one is at Louisville. If you cut down food intake a little bit, like 25%, you have this much of a lifespan extension. If you cut down more, then you see this much of lifespan extension. By the way, this is dependent on strain or species. So do not take these numbers as it is. Now, the thing is, not only mice or rat, pretty much all model organism, traditional or non-traditional organism tested so far, it works. It extends lifespan. It makes animals healthier. So some people actually trying themselves whether it works for humans as well. So my organism, Drosophila, how do we do the experiment? It's relatively simple. Here, we make a semi-defined diet or media, you can say. Major components are sucrose and yeast. Sucrose is carbohydrate source. Yeast is protein mainly, and you have some fats and minerals and vitamins or mixed together. So you can make actually serial deletion from 20%, 15%, 10%, 5%, 1% like this. And this is one of the established protocols in the field. And you can simply say 20% is this type of diet, and 15% is like this. Used to be my diet, but I recently changed from this to this. I feel healthier. So if you measure lifespan of these animals, for example, 15%, suppose this is a relative control. You measure lifespan, this y-axis is a person's survival, x-axis is time by days. So if you measure mean lifespan around here, you can actually see roughly 50 days. If you give more nutrition, the lifespan is decreased like this. Okay, like this. However, if you dilute the food like 10%, for example, then lifespan is extended. If you dilute more, for example, 5%, even more, you see lifespan extension. However, you cannot do that forever because at a certain point, malnutrition starts, so lifespan is reduced. So in this particular case, roughly 5% sucrose and yeast are the best in terms of a lifespan modulation or extension. Now, my question is whether circadian clock is important for this phenotype. Suppose if you remove circadian clock, what happens? Generally, lifespan is reduced or increased, or more specifically, more importantly, whether diet restriction still holds true or not. That is my question. That was my question. So to make a long story short, ISO31 is one of the wild types that people use in the field. As you can see, you see a nice lifespan modulation by different diets. This is blue, this is the best one, and decrease, concentration dependent decrease, and here. But if you measure lifespan of a clock mutants, so CLK, JLK, there's a clock jerk allele of clock gene. These animals, these flies, the mutant flies are behaviorally arrhythmic and molecularly arrhythmic as well. So those flies have completely lost circadian clock inside. As you can see, they do not respond to diet restriction as much as the wild type control here and here. And we did actually multiple trial to make sure this is really the response. And we made a similar observation. And recently, with some undergraduate students supported by the K-Brain grant, we made similar observation with different diet restriction protocol, because one of the reviewers, not the grant proposal, but the manuscript that I submitted, they said this is one particular diet restriction protocol that people do not use a lot these days. So we try different method, or it works fine. So I'm pretty much convinced that circadian clock is important for DR-mediated lifespan extension. One thing interesting is, if you look at malnutrition condition here, 1%, now clock mutants actually outlived. This is quite interesting. So in certain condition, losing circadian clock may be better. So this is clear and great example, I think, of how gene or how genetics interact with environment. to change traits in a certain character. So this is from carried over from my postdoctoral work. So we decided to analyze circadian transcriptome using RNA sequencing analysis. So this is a control food, like a 15%, and this is a diet restriction is 5%. So every 2 hours, we collect the samples and run the RNA sequencing analysis. This is actually quite a lot of work. So using bioinformatic analysis, which I got help from friends at Northwestern. So we found quite interesting gene transcriptome reprogramming. For example, certain genes are only cycling like this in diet restriction condition, while some genes are still cycling in both conditions, but a little bit of different phase or amplitude or something like that. So my question for the grant proposal, not only K-brain and also R15 is first to identify what we call clock-controlled genes. All these clock-controlled genes that show rhythmic expression in mRNA level. What are those functions? What do they do? Especially in terms of lifespan modulation, and also other stress response and sleep and other behavior, including feeding, we recently included here. Second, before that, we recently found one of the cycling genes called RPI, ribose biphosphate isomerase, which is in the pentose phosphate pathway. If you reduce the expression level of this gene, then you see lifespan extension. And we checked other pentose phosphate genes, looked similar. This is quite interesting. So this is one direction we want to go further. And this was done by some undergraduate students in my lab. So second aim is a very specific example of clock-controlled genes. So to make a long story short, using bioinformatic approach from my collaborators, we found some genes, especially in the proteasome subunits. So proteasome subunit, you have a roughly 30 subunits, and proteasome is important for cellular degradation, protein damage, or unnecessary transcription factors after done with their job, or misfolded one. All those can be degraded into in the proteasome complex. So many of these proteasome subunits gain oscillation by diet restriction. It's quite dramatic or interesting phenomenon. So to understand that impact on lifespan, we knock down some proteasome genes and measure the lifespan. As you can see, this is a wild type. You see a nice doses dependent lifespan modulation. Yet, if you knock down proteasome genes, then you don't really see a strong response. So this is a clear indication that proteasome is important for diet restriction mediated lifespan modulation, probably affected by somehow circadian clock. So we're trying to understand the mechanism, how diet interact with some pathway to alter proteasome gene expression. This is a second aim. Third is a bit different, but the conceptual aspect of this aim is still connected to the main topic, which is genetic mechanisms of diet restriction by the circadian clock in Drosophila. Now, we found circadian clock is important for DR-mediated lifespan extension. Now, we are trying to employ a different approach using a specific mutant called PERS. Unlike wild-type flies that have 24 hours of rhythm, these mutant flies, endogenously, they only have 20 hours instead of 24 hours. In fact, this is one of the mutant animals first discovered ever to show the single gene mutation caused behavioral rhythm change. And this later discovered was actually used by Nobel Prize winner 3 years ago. My advisor's advisor, this led to Nobel Prize to them. So my hypothesis is that if you change their rhythms, that being said, if you mismatch endogenous rhythm and outside rhythm, what happens to the diet restriction response. So in wild type, we know that if you put them in 12 hours and 12 hours, if their circadian clocks are aligned inside and outside, you enjoy lifespan extension. But what happens if you put them in different, for example, 10 hours and 10 hours cycle? In that case, they're misaligned. And per S case, they have a 10 hours and 10 hours. If we put them in 10 hours cycle, they're aligned. So probably they will extend lifespan. But in 24 hours case, they're misaligned. So what happens to their lifespan? So this is, I believe, a short and sweet study that can be easily done by undergraduate students. So my reviewers still half and half. Some people like it, some other people didn't like it because this is a little bit deviate from the first aim and second aim. I'm still kind of debating myself what's the best. So we submitted R15 and two major issues. They said the PI, which is to me, is isolated. What that means is that in Kentucky, Louisville area, the PI is the only fly lab. So he cannot get help if he has any problem. That is true. That is true. So I'm trying to form like a fly group in Kentucky, like a KFC. We have several actual labs, Western Kentucky, Murray, and also UK as well. So probably you can help each other conceptually, or we can share reagents, share mutants or something like that. And we can have a short meeting at annual KS meeting, like 1 hour of fly meeting or something like that. So I think I can show that to the reviewers, say that I am getting help from local fly researchers. Second is PI's productivity is a little bit mild, which is true. So these papers in line. We just finished writing and proofread and reviewed by ex-advisors. We're trying to submit a review paper to submit. And another paper from my graduated master students. So we're trying to submit a short paper. There's basically some connection between sleep and feeding. And my postdoctoral work that has been delayed for many reasons. So one reason is I do not have much control. All this first one, second one, I'm the PI, so I can decide myself. My postdoctoral work is my advisor is kind of still hesitating in terms of direction. So once this is done, probably I might have a better chance in terms of score. Before I finish, this work is connected to funded research, but a little bit different. So for example, I'm trying to understand how feeding behavior changes over time, and also how feeding behavior is connected to sleep and circadian rhythms. So these are selected flies. As you can see, each line is independent. Some lines, they sleep a lot, while some lines sleep less. If you measure the food consumption, people might say something, but this is not clearly understood. So I see a very nice inverse correlation between feeding, food consumption, and sleep pattern. So if flies sleep less, they eat more. And if flies sleep more, they eat less. So it's clear inverse correlation. I got some RNA-Seq voucher from Genomic Center. So I'm planning to sequence some representative lines to understand how and what genes are actually co-regulating these two phenotypes at the same time. Another thing, this is, I believe, the last slide. I'm also interested in doing genetics of food consumption. So we're using GWAS, genome-wide association study. So we have a wild type population, which is from outbred, but inbred many years, and each line is fully inbred, and they reach it homozygosity, meaning that there's no variation. So genetically identical each other. And these all lines are fully sequenced. So this is freely available in fly community. So my question is, can I find any genetic interaction or loci that determines food consumption, especially in different sugar consumption? So low sugar and high sugar, you see different response. So preliminary data by my graduate student and some undergraduate students, we found some potential candidate loci. We're trying to understand whether these genes or loci is important for this feeding behavior. This is a long-term project. Hopefully, this can be another proposal. All right, with that, I would thank my lab members and my postdoc advisor and the collaborators at Northwestern. And thank you very much for your support. This is really, really great. This is really saving me. Right. Before I finish, I'm willing to collaborate for any type of research. Drosophila is really good for functional screen. You can do quickly. Really interested in aging. If you do not have time to measure lifespan, I can do that for you. And I measure different behavior like sleep, circadian, feeding, other things. So if you have any interest at all, please let me know. We can work together. All right. I'm happy to take any questions.
So as you were showing your slides and you have DR for diet restriction in my field, because I work on the visual system, that usually stands for dark rearing. So I was just kind of curious, have you ever just done, no light dark cycle at all, just dark reared fruit flies? Does that change their?
Yes, interest. Yeah, that is actually, remember the circadian clock inside, if you put them in constant condition, they lose, especially constant light. So constant light, they reduce lifespan a little bit. And also I have old data that also reduce DR mediated lifespan extension that also suggests circadian clock is also important. If we put them at dark, I do not think that has significant impact, however. So constant light has a negative impact.
Okay, cool. Any other questions? Or, I mean, we can discuss any of the presentations. Well, if not, I mean, I think, this is all going to be on the website, and I hope that you guys all, continue to discuss your projects through email or whatever. All right, thanks so much, everyone.
Thank you.
Thank you.